Namaskar. Welcome to the session on trends in food microbiology. Coming to the introduction. Food microbiology explores the fundamental factors affecting the presence, activity, and control of microorganisms in food. The subject also deals with the crucial concepts required to meet the minimum standards for food safety. Food microbiology includes a wide range of concepts which include food production and processing using microbes, for example cheese, beer, yogurt, wine, etc. Study of microbial induced food spoilage and detection and prevention of foodborne diseases. In the recent years, a drastic shift has taken place between the use of simple microbial plate count to the use of molecular biology techniques to assess and evaluate the quality of food. This shift is essential to live up to the changes that occurred in food industry, evolution of pathogens, and the change in people's behaviors and susceptibilities. This module is divided into the following sections and subsections. First, basic food microbiology, which includes brief historical perspective, factors affecting the growth of microorganisms in food. Second, the microbiological criteria, which includes the indicators of microbiological quality, and the Food Safety and Standards Authority of India, which is FSSAI. Third, culture methods used in food microbiology, that is the quantitative methods and the qualitative methods. Fourth, rapid and advanced techniques in food microbiology, which includes the types of rapid techniques, the advantages and limitations of rapid and advanced techniques, and the recent developments in detection techniques. Basic food microbiology. Brief historical perspective. Following the discovery of the ubiquitous existence of microorganisms by Anton von Leeuwenhoek around the 1670s, scientists started exploring the possible association of microorganisms with food spoilage, food fermentation, and foodborne diseases. Louis Pasteur is credited with the major development of ideas on the possible roles of microorganisms in foods. This paved the way for the establishment of early food microbiology in the 20th century. Some of the major developments in the field of food microbiology are briefly listed in Table 1. Affecting the growth of microorganisms in food. There are two primary factors that affect the growth of microorganisms in food products, that is intrinsic and extrinsic parameters. Intrinsic parameters are the properties that exist as part of the food product itself. Examples of intrinsic parameters are pH, moisture content, oxidation reduction potential, nutrient content, antimicrobial constituents, and biological structures. Extrinsic parameters are the properties of the environment, that is processing and storage, that exist outside of the food product, which affect both the foods and their microorganisms. Examples of extrinsic parameters are storage temperature, relative humidity, presence or concentration of gases, presence or activities of other microorganisms. Microbiological criteria. Microbiological criteria are used to differentiate between an acceptable and unacceptable product or food processing and handling practices. Microbiological criteria are used to assess the following: adherence of good manufacturing practices, that is GMPs, the safety of food, the maintenance of quality or shelf life of certain perishable foods, and the suitability of food or ingredient for consumption. The main objective of microbiological criteria is to ensure the safety and quality of food. Microbiological criteria are an underlying component of any critical control point that address a microbiological hazard in hazard analysis and critical control point, that is HACCP systems. Indicators of microbiological quality. Indicator organism can be used to determine either the microbiological quality or safety. Indicator organism are the indicators of quality. The indicator organisms of food quality should fulfill the following criteria. Should be present and detectable in the food where quality is being assessed. The growth or numbers should share negative correlation with product quality. Detected and enumeration should be easy and within a short time. Clearly distinguishable from other organisms. Their growth should be inhibited by other background microflora. Indicator metabolites. In certain cases, a correlation is established between the presence of certain metabolic products and product quality loss. In these, the microbiological quality is determined by assaying the metabolic products. The indicator organisms or metabolic products correlated with product quality are summarized in Table 2. Indicator microorganism, indicators of safety. Safety is determined by the presence or absence of pathogenic microorganisms and their toxins. Some of the most prominent foodborne pathogens include Salmonella, that is the multi-drug resistant strain, Campylobacter jejuni, Escherichia coli 0157H7, Listeria monocytogenes, Staphylococcus aureus MRSA, Vibrio vulnificus, Yersinia enterocolitica, Archobacter species, and Mycobacterium paratuberculosis. The Food Safety and Standards Authority of India, FSSAI, was established under Food Safety and Standards Act of 2006, which consolidates various acts and orders that have hitherto handled food-related issues in various ministries and departments. FSSAI has been created for laying down science-based standards for articles of food and to regulate their manufacture, storage, distribution, sale and import to ensure availability of safe and wholesome food for human consumption. The FSSAI is responsible for protecting and monitoring public health through the regulation and supervision of food safety. Conventional methods for detection of microorganisms in food. The conventional methods employed for the microbiological evaluation of food and food ingredients can be broadly categorized as quantitative and qualitative methods. Quantitative methods are devised to enumerate indirectly or directly the microbial load in the test material. Conventional culture-based methods rely on specific culture medium for the isolation and enumeration of bacterial cells in 5 steps, which include pre-enrichment, selective enrichment, selective plating for colony identification, biochemical presumptive tests, and serological confirmation. Plate count method. The standard plate count or aerobic plate count APC method is commonly used to enumerate the total number of microorganisms in food products by determining the colony forming units, that is CFU. Most commonly, aliquots from selected dilutions are either pore plated or surface plated on either non-selective or selective agar medium or non-selective or selective differential media. Drawbacks of plate count method include: this method measures only live cells and therefore would not be of value to determine the quality of raw material used in heat processed food. Certain bacteria, though present in low counts, may be able to induce preponderant quality loss due to enhanced biochemical activity. This method is of little value in evaluating the organoleptic quality of food. Direct microscopic counts, DMC. This method is used to give an estimate of microbial cells in samples either by counting stained cells under a bright field or live cells under a phase contrast microscope. The counts are expressed as microscopic counts per milliliter or gram of food sample. Drawbacks of DMC method are: the sample should contain relatively large number, that is greater than 105 colony forming units per ml of microorganisms for effective use of this method. It is not recommended to differentiate between live and dead cells, as it requires the use of fluorescent dyes such as acridine orange. Cannot effectively enumerate microorganisms in foods that have particles. Next is the most probable number, MPN. Aliquots from serially diluted samples are inoculated in broth in tubes containing selective agents, one or more, and incubated at recommended temperature and time. The broth tubes in each dilution are then scored for the presence and absence of growth. The number of tubes displaying growth in each of the 3 successive dilutions are correlated with the number of viable cells of the specific microbial groups using the available statistically calculated table. Dye reduction test. This test relies on the principle that some dyes, for example methylene blue, are colored in oxidized states but become colorless in reduced state under the influence of microbial growth and metabolism. The assumption is that the rate of reduction of the dye, that is the specific concentration added to food, is directly proportional to the initial microbial load of the sample. Qualitative methods are devised to determine whether a representative amount of food sample or a particular batch contains a particular bacterial species which is generally pathogenic among the total microbial population or not. Isolation of pathogens. This method aims at determining whether a particular sample does or does not contain spores or viable cells of specific pathogens. Some of the pathogens tested include Salmonella species, Shigella species and Vibrio cholerae. This is normally considered as only a presumptive test which has to be followed by confirmatory test. Test for toxins. Some pathogenic bacteria, for example Staphylococcus aureus, produce toxins in the food they grow, causing food poisoning upon consumption. Microslide precipitation method is performed in which the extracted and concentrated toxin from the food sample is assayed against specific antibody. Rapid and advanced techniques in food microbiology. Owing to the fact that conventional methods are labor intensive and lengthy, different rapid automated methods have been developed in the recent years to detect microbial load, foodborne pathogens and their toxins. In food microbiology, rapid methods include the newly developed techniques for detection of microorganisms or any modifications introduced into the conventional method that ultimately reduces the analysis time. The advent of biotechnology has enormously altered food testing methods, and numerous assays that are specific, faster and more sensitive than the conventional methods have been developed. Types of rapid techniques. The common types of rapid advanced methods are modification of conventional microbiological methods, nucleic acid based detection, antibody based detection and biosensor based methods. Coming to the modification of conventional microbiological methods. This category comprises of any alteration or modification of conventional assays that result in saving labor, time and materials. For example, Petri film test. In this test, disposable cardboard containing dehydrated media is used for the enumeration of total bacteria, specific bacterial species or mold and yeast. This test eliminates the requirement for preparing media and agar plates and economizes in storage and incubation space. Chromogenic and fluorogenic substrates. The introduction of chromogenic and fluorogenic substrates in special microbiological media provides a quick measure of specific enzyme activities which are characteristic traits of certain bacteria or bacterial groups. Example, chromogenic substrate like orthonitrophenyl beta-D-galactoside, that is ONPG, and 5-bromo-4-chloro-3-indole beta-D-galactoside, that is XGAL, are incorporated in the culture media to measure the activity of beta-galactosidase, that is GAL enzyme. Next is the ATP bioluminescence. This technique measures using a luminometer the light emitted by an enzymatic reaction between luciferin and luciferase that requires the presence of ATP. The amount of light emitted relates to the number of microorganisms. Nucleic acid based methods. Nucleic acid based food pathogen detection assays are highly specific as they detect specific nucleic acid sequences in the test organism by hybridizing them to a synthetic oligonucleotide complementary to the specific nucleic acid sequence. Numerous nucleic acid based assays including hybridization, amplification, microarrays and biochips have been designed for use as rapid methods to detect foodborne pathogens. Polymerase chain reaction, that is PCR based methods. Simple PCR method. In this method, double-stranded DNA is denatured into single strands and specific primers are annealed to these DNA strands, followed by extension of the primers complementary to the single-stranded DNA with the Taq and other thermoresistant DNA polymerases, figure 1. The quantity of the products of amplification can be visualized as a band on the ethidium bromide stained electrophoresis gel. Simple PCR methods for toxin detection have been developed for a range of bacterial species such as Bacillus cereus, Vibrio cholerae, Staphylococcus aureus and Escherichia coli. Multiplex PCR. The method of rapid detection of multiple microorganisms in a single reaction by simultaneous amplification of more than one locus is referred to as multiplex PCR or MPCR. Example, MPCR assay for the simultaneous detection of Salmonella species, etc. Quantitative PCR, qPCR, also called as real-time PCR, is a system competent of continuously monitoring the formation of PCR amplification products throughout the reaction. It offers simultaneous rapid amplification and sequence-specific based detection of genes of interest and is progressively being applied in food microbiology. Fluorescent in situ hybridization, FISH. FISH with oligonucleotide probes targeted a tRDNA is the most common method among molecular techniques not based on PCR. The probes used in FISH are generally 15 to 25 nucleotides in length and are covalently labeled with fluorescent labels or radio labels with P32. After hybridization, the specifically stained cells are detected using epifluorescence microscopy or autoradiography. FISH in combination with flow cytometry has been employed for rapid culture-independent detection of Salmonella species on the surfaces of fresh produce like tomato. Isothermal amplification. In the recent decade, many novel methods have been developed to amplify nucleic acids under isothermal conditions. These methods include nucleic acid sequence-based amplification, that is NASBA, loop-mediated isothermal amplification, that is LAMP, strand displacement amplification, SDA, and rolling circle amplification, RCA. Isothermal amplification has simpler, cost-effective hardware requirements compared to PCR. Antibody-based detection. Immunological detection based on the concept of antigen-antibody binding is widely used for determining foodborne pathogens. They are as follows: enzyme-linked immunosorbent assay, ELISA. One of the most commonly used immunological assays, for which is a very accurate and sensitive method for detecting antigens or haptens. Binding of antigen, that is pathogens or toxins, to the primary antibody is measured quantitatively in a 96-well microtiter plate by using a secondary antibody conjugated to an enzyme. The most powerful ELISA format is called the sandwich assay because the antigen to be measured is bound between two primary antibodies, the capture antibody and the detection antibody. Biolin Salmonella ELISA test for Salmonella species is a rapid, easy and convenient assay for the detection of Salmonella in foods and feeds. Immunomagnetic separation, that is IMS assay. This procedure, which utilizes immunomagnetic beads, that is IMBs, are capturing reagents, is designed for microbial isolation and identification. IMS is analogous to selective cultural enrichment, in which the growth of other pathogen is suppressed while that of the target pathogen is enhanced. The separation is a two-step process. First, the target cells are combined with immunomagnetic particles, incubated for about 1 hour and separated by a suitable magnetic separator, and second, the magnetic complex is washed repeatedly to remove the contaminants. Lateral flow immunoassay. Lateral flow assays are a type of immunoassay where the test sample flows along with the solid substrate via capillary action. After the sample is applied to the test, it encounters a colored reagent, that is antigen or antibody labeled by gold particles or colloidal or gold particles, which mixes with the sample and transits the substrate in the process, encountering lines or zones that have been pre-treated with an antigen or antibody. Depending on the type of annihilates present in the sample, the colored reagent can become bound to the test line or zone. Lateral flow immunoassays such as immunochromatography, dipstick and immunofiltration are garnering attention in the detection of pathogens and toxins in food. Examples include immunoassay based lateral flow dipstick for the rapid detection of aflatoxin B1 in pig feed. Biosensor based methods. Biosensor is an analytical device that comprises of two main elements, a bioreceptor and transducer. The bioreceptor that recognizes the target annihilate can either be a biological material, that is antibodies, enzymes, cell receptor and nucleic acids and cell receptors, or biologically derived materials such as recombinant antibodies and aptamers, or biomimic, that is imprinted polymers and synthetic catalysts. The transducer, which converts the biological interactions into a measurable electrical signal, can be electrochemical, optical, thermometric, mass-based or micromechanical or magnetic. Optical biosensors uses surface plasmon resonance, that is SPR. In SPR, The electromagnetic radiation of a certain wavelength interacts with the electron cloud of the thin metal and produces a strong resonance. When the pathogen binds to bioreceptors which are immobilized on the surface of a thin metal surface, the refractive index changes, which results in the change of wavelength required for electron resonance. Electrochemical biosensors. Electrochemical biosensors measure changes in current, impedance, voltage and conductance caused by antigen bioreceptor interaction and are classified as amperometric, impedimetric, potentiometric and conductometric respectively. Mass-based biosensors uses piezoelectric crystals. The binding of the target antigens to the antibodies immobilized on the crystal, this will cause a measurable change in the vibrational frequency of the crystal. The rapid techniques for the detection of foodborne bacterial pathogens are summarized in Figure 2. Recent developments in detection techniques. Biosensors based on nanofabrication techniques are emerging as rapid detection methods for foodborne pathogens. Quantum dots are being explored as potential fluorescent probes for detection. Micro-cantilever based biosensor for detection of Vibrio cholerae 01 are being explored. The commercial gold-plated atomic force microscopy micro-cantilevers are immobilized with a monoclonal antibody and used to detect Vibrio cholerae 01. Another area where research is being focused is the phages, which can be used for both detection and prevention of foodborne pathogen. Advantages and limitations of rapid detection techniques. The rapid methods of pathogen or toxin detection have the distinct advantages of rapidity, specificity, sensitivity and less samples over conventional culture-based methods. The advantages are summarized below. Results are rapid, reduces both labor and time consumed. Tests are very specific and sensitive, allows detection of specific serotype. The tests and devices are portable, can test for microorganisms that cannot be cultured, allows for earlier detection and faster treatment. However, these techniques are also plagued with certain limitations which include expensive investment in new lab equipments, may require additional technical expertise. AOAC International approved rapid methods are mostly designated for preliminary screening. Negative results are regarded as definite, but positive results are regarded as presumptive and need to be confirmed by standard microbiological methods. Potential for abuse bioterrorism. Conclusion. The field of food microbiology has been rapidly and steadily developing in the past century. While conventional methods to detect foodborne pathogens have primarily relied on culture media, the rapid methods rely on advanced molecular techniques. Recent developments have shown that there are promising tools on the horizon in the form of quantum dots, phage-based assays, and micro-cantilever biosensors. Given the broad applicability and the great potential of such methods, there is still a great chance for further developments.